Okay, good morning everyone. We are EPO Biosolutions, and today we're presenting our project on recombinant human EPO production via microbial culture coupled with synthetic glycosylation. So I want to introduce the team first. So here we have Will, who is our Chief Operating Officer. We have Gabby, who is our Chief Scientific Officer. Unfortunately, she has some kind of medical emergency and cannot be with us today. We have Faisal, who is our Chief Financial Officer, and Bryan, myself, who is the Chief Executive Officer, and our mentor, Emma, who is sitting in the audience. So brief overview of what we will be talking about today. We'll start with the introduction, talking about background of EPO and the technology we're proposing, and then we're going into our base case, talking about business model and preliminary studies. And then we will go into the final solutions, and at the end, a summary slide. So what is EPO? EPO's full name is erythropoietin. It is a hormone produced in your body, specifically your kidney, to stimulate the red blood cell formation in your bone marrow. So clinically, it is used as a therapeutic protein to correct anemia. And currently, Amgen is the sole technology holder to produce this therapeutic protein. As you can see, that glass vial of EPO, it's called EPOgen. Its dosing price is $300 per dose, and patients usually have to take up to 3 doses per week, which is very expensive. So what made EPO this expensive is mostly its manufacturing process. So what does Amgen use as its current manufacturing process? So it is using Chinese hamster ovary cells, which is also known as the CHO cells, to produce this protein. Through the central dogma of biology, DNA to RNA to protein, they would insert a human gene into the CHO cell, and then the CHO cell would then express the protein, and through a series of purification steps and final formulation, we will have the final drug product. So this technology was patented in 1989, but it has recently expired in 2015. The advantage of using animal cells is that they have the necessary machinery to go through glycosylation, which you know is very important for biological activities. However, the downside of using animal cells is they are very high maintenance and they have very slow growth. Compared to a doubling time of less than an hour for bacteria cells, mammalian cells usually have a doubling time of 24 hours. So because of this high maintenance and slow growth, the manufacturing process is usually very expensive. Also, the current process is a fed batch process. So there are some efforts put into the development of new processes, including perfusion process, which is continuous, so it's able to support a much higher cell density, which in turn gives a high productivity. But because this process is continuous and it is very complex and labor intensive, it might not be very cost effective in terms of to reduce the price for the drug. So this has led us to think backward. If animal cells have the necessary machinery to go through glycosylation, will we be able to do glycosylation without this machinery so that we can use bacteria cells, which are way cheaper to use as a manufacturing platform? So there are some recent papers published on click chemistry highlighting the mechanism of conjugating a molecule onto a protein using click chemistry linkages. So we are investigating this technique to use in our process. So we propose this technology of producing EPO from E. coli and use a synthetic glycosylation step to chemically add glycans onto the protein. So doing a quick research on the primary structure of EPO, we found 4 glycosylation sites. There are 3 N-linked glycans and 1 O-linked glycan. And reading from literature, the 3 N-linked glycans are the ones that are really important for biological activities. So we concluded that N24, N38, and N83 are the 3 sites of interest that we need to add the glycans onto the protein. So like any traditional bioprocess, we are starting with a working cell bank and go through genetic modification. So what's new about our process compared to the old Amgen EPO process is we are using a bioreactor to cultivate bacteria. And then we have another series of reactors to go through synthetic glycosylation. And then we will have downstream purification and final formulation to formulate our final drug product. So because the working cell bank, genetic modification, and formulation are very well established, and we're not changing much, our focus is mainly on the bioreactor design, synthetic glycosylation, and purification.
So for the best case, we went into look into the business plan, economic feasibility, and initial process design and preliminary calculations. We decided to go with hybridized model in terms of developing the process. First, we'll start with technology partnering with a company that's already on the market. For instance, Amgen or other competitors trying to get into the biologics. Once we've established ourselves and have garnered some recognition in the market, we'll then try to go into asset creation and out licensing. Eventually, we want to become product development and commercialization company ourselves, like Amgen, but also not just for like the biologics and other drugs too. We'll be using this technology to produce antibodies, hormones, and other stuff, not just proteins. Currently, our mission is to use our knowledge and skills in science and engineering to create innovative platforms for high-quality drug delivery to make medicine affordable for everybody. When we did our market research, we identified these companies as the major players in the market in terms of EPO and what share of the market they control. And our target is to capture only 5% of the global market. By doing so, we'll be able to generate revenue of about $343 million per year. This is a graphic showing the trend of the current EPO market. In 2014, it was mostly biologics, a little bit of biosimilars. But as time goes on with the expiration of the patents, there's a growing trend of biosimilars coming into the market. So that's why this technology that we're proposing is essential, because it will propel the growth of biosimilars and make the overall drug cheaper for everybody.
So knowing that we are targeting 5% of the global market, this roughly correlates to a 600 grams of production rate annually. So by gathering some pricing information from manufacturers for equipment and materials, we did a rough class 4 estimation. So as you can see, for production costs, it roughly came out to be $34 million. And if we price our EPO product at half of its market price, we'll be able to make about $468 million per year. And if you subtract that, it gives us a margin of $434 million per year. And if you assume a corporate tax rate of 21%, we will be able to make a profit of $343 million per year, which definitely proves our process will be viable, which led us to further investigate our technologies.
So after we honed in on our business goals and determined some initial economic feasibility, we began to look into the specifics of the technology itself, starting with the first area of interest for our focus, which was the design of the bioreactor. So considering some different alternative bioreactor designs, our goal was to choose something for the fermentation step that was well established. Ultimately, that led us to look only at batch processes as opposed to higher yield and continuous processes like perfusion reactions. And for E. coli, they're not very shear sensitive bacteria, they're not very delicate. So a continuous stirred tank bioreactor is preferable to an airlift bioreactor among those two popular batch reactor geometries. The medium that we hope to culture E. coli in, LB broth is used in a lot of bench scale work. It's a very well established broth medium, but we're hoping to use a high yield broth which can lead to higher cell densities in a batch geometry and potentially lead to higher protein expression rates as well. And the material of the reactor, we considered using single-use disposable reactors, but because the aeration is a major priority in our fermentation, and because single-use reactors are still an up-and-coming technology, and while they are progressing rapidly, they haven't hit the aeration efficiency of a permanent reactor quite yet. Our plan is to use a permanent stainless steel reactor. This then led into investigating our synthetic glycosylation step, which is the crux of our proposed design. So one idea was to modify the end sites on the protein to cysteine residues and use those to target for glycosylation. The bench scale demonstrations of this we saw in literature weren't very specific, and we thought that it would have a pretty low yield and a high load on our purification systems, so we looked into other alternatives. Copper click chemistry was the direction that we started going into because it solved that specificity issue. However, in talking with our professor and some others, we were advised against it because of the toxicity of copper potentially posing an issue, both for our purification scheme and also for getting this drug FDA regulated. It could potentially pose an issue. So we were suggested to look at DBCO click chemistry, which is, DBCO is not metallic, so it poses fewer issues there. And it's been demonstrated at the bench scale to be specific in the area in which we're looking. This is a simplified view of what will be happening in our DBCO reaction. The DBCO is conjugated onto the protein, and then that occurs in one reactor, and then that is brought into another reactor where the glycosylation occurs on the conjugated DBCO sites. And this is the kinetic data just from the paper, which gave the demonstration of that chemistry.
So we then look into the purification scheme for this process, and we considered two types of cell harvesting. Initially, we thought we were going to use disk stack centrifugation, but then it turned out that for this stack centrifugation to be useful, it has to be above 4,000 liters operating volume. But our operation is not up to 70, it's about 70 liters max. So we then went to use depth filtration. For the chromatography, we considered ion exchange, affinity chromatography, size exclusion, hydrophobic interaction, and membrane absorption. Of all these, ion exchange and affinity chromatography showed the highest resolution. And for our purpose, going to FDA and trying to partner with other companies, we don't want to give any leeway to a product not being pure enough or contain any unwanted proteins. So we decided to go with the ion exchange and affinity chromatography. And we also added a viral filtration at the end to capture anything that goes through these two chromatographic columns.
Taking into consideration all of this, we developed an initial process flow diagram kind of showing all of these units in tandem with one another. You can see that there are filtration steps followed by an orthogonal ion exchange column array going into the glycosylation reactors. After the glycosylation reactors, there's an additional purification step before the product is then stored. And then we do not consider formulation, so that's the end of our process as it stands there. And we also ran some initial hand calculations to determine the scale we're working at in terms of the mass balances for each of these unit operations. So from this initial work, this informed our final solution, in which we developed a detailed process design, ran some simulations, and generated some economic results from that, and also considered some of the safety concerns that would arise with this plant. This is a graphical representation of our final process solution. So there's a genetically modified E. coli strain which is inoculated in a shake flask. This is fermented in a batch fermenter. The cells are separated out in a depth filter, and size is separated in an ultrafiltration system, followed by a chromatography system this yields a purified, unglycosylated recombinant human EPO, which goes into the DBCO reactor, which conjugates the DBCO onto the glycosylation sites of the unglycosylated protein. The glycosylation reactor then takes these DBCO sites and adds the glycans to the protein. There's then an additional set of downstream filtration steps, just in case there's anything that's not specific or any misfires in the glycosylation step, yielding a purified recombinant human EPO at the end. From there, we developed a process and instrumentation diagram for each of our unit ops. This shows the upstream system. So we have a shake flask that feeds into a fermenter, and then that feeds into a fermentation product tank, which holds the fluid from the reaction before going into the downstream purification. And you can see here we've made considerations for things like overpressure scenarios, overlevel scenarios, and air filtration, which is taken into account in all of the process and instrumentation diagram. That then feeds into the depth filtration tank, followed by the ultrafiltration tank. And then it goes into a set of 2 chromatography columns to complete the upstream purification, the upstream downstream purification. That then feeds into the glycosylation reactor array, which then feeds into a column and a viral filtration tank for the final product at the end there.
So we used the SuperPro Designer to model this whole process. And as you can see here, this from the cell bank coming into the depth filtration, we go into the holding tank for polishing, and then into the ion exchange chromatography column. From there, we go into the DBCO and then the glycosylation tank, and then come into our finished chromatography, finally to a viral filtration tank. And then we go into the product being frozen in a 50-liter bags. This is a screenshot of the products that we get. This is our main target product, the EPO. It shows it's not very legible here, but we are supposed to be making about 401 grams of our main product per year. And that is within our spec, like what we wanted to make between 300 and 600 grams per year. So it's within that range. And based on that, we plan to send our plant at Durham, North Carolina, because of the cheaper taxes and also availability of other biopharmaceutical companies in the area. And also our initial investment is estimated to be at around $13.3 million, annual operating cost of $13 million, and taking all this into account, out of our revenue of $357 million, we'll probably be making about $272 million in profit at selling our product at 56% of the current market price, also considering 21% tax rate. After the third year of our operation, we'll have an internal rate of return of about 370%.
So Will has talked about the control system in the P&ID slides. So how are we actually implementing this control system into our process? So first of all, we want to operate our process in a clean room to be CGMP compliant. And this also prevents contamination from the environment to our plant and the contamination of our plant to the environment. Also, all our uniops operate within the temperature of room temperature at 37 degrees Celsius. So this is well within the operation limits of the equipment. The pressure is just normal 1 atm, so also within all the operating limits of the instruments. We have fail-open draining valves to prevent overflow of the tanks, and fail-open relief valves to prevent overpressure and tank explosion accidents. Moreover, we have air filtration outlets on all the uniops, so this prevents toxic particles to release from the reactors or any other holding tanks. So next step for us would be to have the EPO we produce to be approved by the FDA. So our EPO is considered a biosimilar compared to EPO gene because it is mostly identical, except for between the glycan and the actual protein, we have a DBCO and enzyme linkage. However, there's no papers that ever suggested that this linkage is toxic in vivo, so we really expect there's a high chance that this product get validated, but we do anticipate a 6-year approval period, which includes IND filing, phase 1, phase 2, and phase 3 of the clinical trials, and we expect to spend around $100 million for the approval process. However, once our first product is approved, we will be able to expand our manufacturing platform over a variety of biological products. This can include hormones, antibodies, enzymes, and a lot more, which will enable us to truly make medicine affordable for everyone. So in summary, for our project, we are producing recombinant human EPO using E. coli batch fermentation process coupled with synthetic glycosylation. Our business model is technology partnering, and we are targeting 5% of the market share, which correlates to around 400 grams per year. And with that, we project a profit of 272 million per year. We want to thank our mentor, Emma Chori, who's sitting over there. And we want to thank the Northeastern Chemistry Department, especially Professor Ron Whaley for getting the software Super Pro designer to be working. We want to thank Professor Bentry, Fluger, and Webster for organizing this course and giving us the opportunity to present here today. And we'll be taking any questions.
First, what is your team number, Rick?
Number 4.
Any questions from the audience?
Could you look at the business model you had, the second slide or so? The model you're using, it seems to me that you would be licensing later in the process.
Yes.
Proving, not just proof of concept, but market products, product efficacy, economics, and those things. That's how you build value in a licensing business. Here you're licensing something before, really early in the process, diminishes the value. And as they say in the licensing business, the worst thing that ever happened is you get one license that you have to support. So I would have thought differently about that. I would have staged it differently. Short term, who would you partner with? Did you consider a partner? Obviously you considered a partner. A specific partner?
Amgen, for one.
Okay.
J&J, Janssen.
And also a lot of the small companies who are trying to enter the biosimilar market.
I think that this is also after, this is our commercialization plan after, the proof of concept work and having a pilot demonstration of our technology before we're going to be approaching industry partners to invest in this process, basically. So while this is short term, this is, I guess, short term in terms of our commercialization timeline.
Other questions from the audience? So for the ultrafiltration vessel that you mentioned at one point, is there any precedent for that? Or is this something you guys are developing?
Which slide is it?
It's this one here.
This?
I think there's an ultrafiltration vessel.
The ultrafiltration.
Oh.
Ultrafiltration. I think it's this one.
Maybe this.
I believe if you go back to the P&ID.
Here.
Yes. So the ultrafiltration system.
Oh, so that's supposed to capture any other inclusion bodies and other proteins that we don't want?
How does it work?
It's pretty much identical to the depth filter, but it's more specific in terms of what it lets through.
So what kind of filter does it use for the ultrafiltration? Do you guys know, or?
I looked it up. It's sized about 2 microns. I'm not sure exactly. It's not I can look it up if
Yeah.
later on.
Just the 2 micron is not ultrafiltration.
Okay.
Ultrafiltration is in the nanometer range.
Hmm.
So you're separating bacteria from protein. That's what you're doing there, right?
Yes.
So your suggestion is that would be too small of a pore size for this process
Too
then?
big.
Too big.
Oh, too large, okay.
Okay, and I'm also intrigued by your chemistry. Did you guys do your homework on that one, or?
Well, Gabby is the one who worked on it most, and she's not here today.
Okay.
But we can try to answer questions.
So, all right, okay. Now, what I want to know, the EPO, human EPO has a certain set of carbohydrates attached to it, right? That makes it humanizing.
Yeah.
So each animal has a different set of carbohydrates. And where are you going to get those carbohydrates?
There are vendors that sell the pre-made glycans.
So
And
just
we
buy
were
those
able to find
glycans and you,
Yeah, if you go back to,
there was one reaction, one process in your process where you actually taking those carbs and just connecting them to your protein. So there are multiple sites for glycosylation, right?
Mhm.
And they don't all have the same glycans, do they?
for these 3 unlinked glycans, they all have the same exact carbohydrate branch, which is the one shown on the right.
Okay.
And we were able to find vendors who sell this pre-made glycan.
Okay, so that you're lucky because generally
Yeah.
proteins have different sets of glycans on the surface. So in this case, you have 3 glycans that are identical. And is there anything else that is not?
Well, the O-linked glycan is different than the unlinked glycan, but we're not considering that because the paper said that site is not very important for biological activities, so it doesn't really affect the in vivo activities for product.
Yeah. And my last question is about the 5% market share. So what is your competitive advantage?
By looking for 5% and selling at half the price, we hope to entice other companies to,
Are you cutting the price by half or something like that? Or?
yes.
And that's our hope as well. Is we chose 5% as kind of a good percentage of the market to break in, to kind of entice like a single manufacturer at a time, as opposed to trying to scale up this process to meet like 50% of the world need and saturate the market and potentially fail
You also
Capstone.
need a big investment up front if you're
Correct.
50% of the
So
market.
all of that would start to scale up. So we decided it was a good break-in. It was a good compromise in terms of breaking into the market.
So what is it going to cost you to launch this project?
I believe the upfront investment to get ourselves up and running is, I think the upfront investment was about 13 million, something like that.
Yes,
Yeah.
about 30 million.
That's
30.
1-3.
Yeah. So that's to
Not
get
sure.
ourselves like running, and then the regulatory process will obviously add a substantial cost to that.
Yeah, it's $100 million or something.
Yeah.
Yes.
Great.
Which will require definitely some venture
Big players.
investment.
Licensing. You need to
Yeah.
license it to a company
No.
to get that kind of money.
Mm.
Very ambitious, but totally feasible.
Only this that.
Well, with your profit projections, I would have been inclined to look at lowering the price 90%.
If they don't care about yield, yeah.
I understand, but, the numbers like that can attract $100 million of capital,
Yeah.
and would. So if you're looking for a competitive barrier entry, a good competitive barrier entry is pricing that would diminish it on the upfront, and then ride it up, and then as as it is successful, the value of your license. You can structure a license lots of different ways, I'm sure you know, but doing it based on production, as opposed to a paid-up license or something like that. With the price rising up is where you'd make the money. You wouldn't make it make a product.
Yeah, there's definitely a lot more room to do optimization on our pricing and economic analysis.
Yeah.
Well, if I was an investor and I put in $100 million for the clinical part, I wouldn't want you to sell the product at a half of the actual price. I want you to have huge margins. Because I want to make more money on my investment, right? So you may want to cut the price by half, but the investors are not going to let you do that. Or they may not let you do that. So you got to have other carrots in your,
Basket, yeah.
to entice them to put in a cabinet, and also to be competitive. So you really have to cut your cost. Really, somehow you have to cut your cost more than what's out there. But actually selling at half the price might not be,
Way to go.
enticing for investors.
Sure.
Yeah. Are there any other questions?
Did you do a net present value on over?
We did, but it's not on this slide. That's why we came up with this number after doing the net present value.
Over 10 years?
Yes.
That is an eye-popping internal
Rate of 10 years.
Terry are way cheaper than a million cells, so.
Any other questions?
No, I think we're done. Thank you.
Thank you.